We're gonna start by going over the warm-up, which is what we always tend to do. So let's do that. We go bring up warm-up. And I need to share that. I'm gonna share. Okay. What that's not right. Stop. Let me share this differently. Yeah. There. That is right. Okay. So yeah, and make sure not everybody got this completed. You always want to have the warm-ups completed before we start the class. The whole point is get them done, and then we're going to go over them. And again, this is also, they're not worth that many points, but they are designed to kind of give you a little kick to make sure you're keeping up with the material and reviewing the stuff we've already gone over, so you're not falling behind. In this class, if you keep kind of waiting to get caught up, you're just going to end up sliding, looking at Ethan's thing, you'll end up below the waves and just, that student sleeps with the fishes. So you really need to keep up in this class, or else it's not going to be possible to claw your way back up to the surface. All right. Oh, not moderate. Dagnabbit. Hold on. We want to go quiz statistics. And like I've mentioned last time, I'll mention again, if you find yourself down in this lower area on these warm-ups, it's not going to hurt you in terms of the overall points because it's a fraction of a point, but it is information like, yikes, I better review this material because we're going to just be building on this and I don't got it at this point. So, all right, so here we have, what is this? This is a classic metabolic pathway here. This is the Krebs cycle, which we're going to look at in much more detail when we do cellular respiration. It's happening in the mitochondria. This is a key part of making ATP from the glucose in your cell. And what do we call, as we're going along here, there's citrate, then there's isocitrate, then there's, so at each stage, there is some other molecule that's getting formed, some other compound that then gets turned into another compound. It gets turned into another compound. What do we call each of these compounds that's not our final product, but that gets produced along the metabolic pathway?
Intermediate.
Yeah, those are all the intermediates or the intermediate compounds. And then what is driving the transition from one intermediate to the next intermediate? What has to be helping this reaction go from one step to the next step?
Enzymes.
Yeah, there's going to be a particular enzyme that is associated with each step of this metabolic pathway. So metabolic pathways are going to be, constantly coming up in our class. So kind of understanding that basic idea and understanding the terminology. Intermediates are the compounds being produced at each step, and understanding that there is some enzyme that is going to catalyze the reaction from one step to the next step. Okay. Oh, then I want to turn this off here. Describe the function of enzymes. So this, a lot of people got the basic idea, but a lot of people didn't get both of the kind of defining properties. Like when you talk about an enzyme is an organic catalyst. And what are the two things that are the defining properties of a catalyst?
They speed up a reaction.
Okay, one is they speed up reactions.
And it doesn't change you.
And it doesn't get used up. It's not consumed in that process. You know, there's lots of other things, but those are like the two real defining properties, you know, and because the catalysts that we're going to be looking at, these organic catalysts, enzymes, are proteins, they're going to be affected by denaturing and this and that. But at the core, catalysts speed up reactions and are not consumed in that process. This one I was a little bummed. We talked about this a few times now. It's clear that people are still not exactly sure what a mole is. 1 mole, does it weigh exactly the same as 1 mole of another element? Most definitely not, right? A mole of hydrogen like floats up into the air. A mole of lead is going to break your foot if you drop it on there, right? A mole is just the amount of atoms or molecules, right? It's 6 times 10 to the 23rd atoms or molecules. Again, a number, like a dozen eggs, right? If I had a dozen bowling balls, that would weigh a lot more than a dozen pom-poms, right? It's the same thing. A mole of helium floats, and a mole of iron definitely does not float. So make sure you understand what a mole is. If you were one of the ones who wrote true here, kind of go back and review what the heck is a mole. And it's going to actually come up on this homework that we're assigning today. Make sure if you're answering the question, what is a mole, that you're not just cutting and pasting. Make sure that you're letting the answer percolate through your brain, so you're writing something out that makes sense. Sometimes people will like answer a question about a definition, but they'll just cut and paste the definition from someplace and actually won't understand what they're writing. So make sure mole makes sense to y'all.
Isn't the mole the atomic weight, or is it the mass?
If you want to actually, the atomic weight is the grams of a particular thing that corresponds to 1 mole of that particular thing. So the definition of a mole, you could say like, you know, a mole of carbon is 12 grams of carbon, but the way to really understand what a mole is a number of things. There's some definitions of mole that says the same number of atoms or molecules as there are atoms in 12 grams of carbon, which is a mole of carbon. But in general, you should think of a mole kind of the same way you think of a dozen, as a number of atoms or molecules. This one, most people got, only one person. When the concentration of hydrogen ions increases, you know, hopefully, yeah, we all saw pH goes down. That was the whole point. Adding the hydrochloric acid, we saw that the pH was going way down. And finally, so this one, hope if you were one of the respondents who said need more information, you also need to kind of think more about osmolarity and dissociation here. You know, when specifying a solution of NaCl dissolved in water, so osmolarity compared to molarity. So we know NaCl is going to break apart into 2 separate independent ions. So we know that osmolarity is always going to have to be double whatever the molarity is. However many moles of NaCls we got twice that many particles, because we have an Na and a Cl independent for every NaCl that went in. Natalia raised her hand. Yes, question?
So is that only when it's like an ionic stuff, or is that always with molarity? But because it's splitting, that's why it's doubling, or is that only because it's sodium chloride's ionic?
I mean, it has to do with the fact that it's ionically bonded and it dissociates. It has to do with the fact that that particular compound dissociates. So like a mole, what about if I was comparing the molarity of glucose to the osmolarity of that same solution? What would be the relationship?
It would be the same because glucose isn't ionic, right?
Exactly, because it does not break apart. Every glucose molecule is just in one particle. So osmolarity and molarity for a glucose solution will be equivalent always. But if it's ionically bonded, like salt or potassium chloride or calcium chloride that we saw in the lab, it will be greater osmolarity. You know, it'd be like 3 times as high if it was calcium chloride, because calcium chloride breaks into 3 ions, a calcium and 2 chlorides. So any questions about any of these?
I have a question. The one that we were just on, does the NaCl have to be in a solution for its osmolarity to be higher than its molarity?
Right, osmolarity is referring to a solution, right?
Okay.
Right, it doesn't make sense in terms of a crystal or anything. It has to do with how many, the moles of particles in a solution, you know, per liter of solution. Yeah, when salt is just in a crystal, then it doesn't make sense. It's just like in a little crystal. Other questions about this? Again, over the, you know, the coming week, we are gonna, actually coming weeks, we're gonna be spending lots more time looking at relationships between molarity, osmolarity, and things like that. They're gonna be critical points. And again, in lecture, I went over just that derivation of why an IV flesh syringe is 0.9% saline, you know, because that is that 0.3 osmolar, that is that physiologically isotonic concentration that's going to make all your cells happy. It's going to make, it's the same osmolarity as the inside of your cells, so the cells don't absorb or lose water. All right. So what we're going to do now is hopefully finish up our chemistry review and finally like start getting into physiology. But we still have a little more chemistry review to do, namely kind of go over the 4 basic kind of biomolecules, like the carbohydrates, the lipids, the proteins, nucleic acids, and then there's also a hodgepodge of combinations of them. But it is important to have a basic understanding of the basic molecules and their properties, since pretty much everything we're going to look at is going to be made up of one of these kinds of things. So let's take a look at basic biomolecules. Okie dokie. So, like I said, there's like carbohydrates, right, which is going to be like sugars and starches and stuff like that. There's going to be lipids, which are the fats, oils, as well as some of the more interesting, the main things that make up your cell membranes, like the phospholipids and cholesterol and the steroid hormones, things like that. Proteins, which make up the enzymes we've talked about, also make up a lot of the structural stuff in your body, like the collagen. That's some of the main kind of just building stuff, and also a lot of the specialized things that are on the membrane to act as receptors and channels. We're going to see proteins doing so many different kinds of jobs in the body. And you know, the DNA, the RNA, and then there's other things like I mentioned last class, like, you know, NADH, that high energy electron carrier. So some of the nucleic acid based things are doing other jobs as well as in addition to carrying information and things like that. So they play a few different roles. So let's just take a look at each of these and just think about their basic properties. We can start with carbohydrates. A defining property of carbohydrates is they're always made out of carbon, hydrogen, and oxygen, and it's always in a ratio of 1 to 2 to 1. In fact, that's where they get their name, right? When people were first looking at this stuff, they're like realized that the ratio of these elements was 1 to 2 to 1. So it's kind of like, oh, carbon and like H2O kind of, so, hydro is water. So it's like, it's not really a carbon and a water, but it's got the ratio of atoms as if it's carbons and waters. So that's how it kind of gets its name. There are 3 main kind of subclasses within here that you should be aware of. Monosaccharides. These are like my simple sugars. This is like glucose. This would be, for instance, C6H12O6. This is glucose. This is also fructose, depending on how you put it together, or it could also be galactose. You can rearrange those C6H12O6s in different ways to make different monosaccharides. For this class, you don't need to know the difference between a glucose and a fructose or something just by looking at it. But you should know that glucose and fructose are simple sugars. This is what we'd call a hexose sugar because it's got 6 carbons. Other monosaccharides have the same ratio but have 5 carbons. C5H10O5. What are examples of this? Would be like a pentose sugar, right? For 5. This would be a hexose sugar for 6. What is an example of an important pentose sugar that you all are familiar with?
Ribose?
Yeah, ribose. Ribose and deoxyribose, the things in DNA and RNA. Deoxyribonucleic acid, DNA, gets its name because in that backbone you have these 5 carbon sugars, deoxyribose. So these are simple sugars. Again, we will be seeing a lot of glucose because it is, the main thing that feeds into cellular respiration, driving kind of the energy production in your cells. The next category that we're going to add in here are disaccharides. Di for 2, it's when you add 2 monosaccharides together. These guys, when you look at one of these, when they are in solution, they make this ring form where there's basically, there's an oxygen, so when I'm drawing my like sloppy drawings of monosaccharides, I'll draw them as like little rings. Disaccharides, you have 2 monosaccharides connected together. these are particularly important because these are transport sugars in plants. These are kind of the main foods that we're getting our sugars from. Like for instance, sucrose, CNH pure cane sugar grown in the sun. Sucrose is a disaccharide that is a glucose connected to a fructose. So when you are having table sugar, you're basically having a glucose connected to a fructose, which then you are going to ultimately break apart to get the monosaccharides before you feed them into the machinery of cellular respiration. Another important one, lactose. Lactose, milk sugar, is going to be a glucose and a galactose. Right? In order to digest milk sugar, you have to break apart that glucose and that galactose. It takes an enzyme, it's what's it called, beta-galactosidase. Some people got that enzyme after they grow up, some people don't. That's going to make a big difference on whether or not you're lactose intolerant, right? If you have the enzyme in your body that can break apart the glucose and the galactose, then you're golden in terms of digesting milk sugar. If you don't have that enzyme that can break apart the glucose and the galactose, then that continues down into your digestive tract where bacteria have no problem breaking it apart, and you feel really crappy because they're like breaking it apart and making lots of gas and things down in there. So, breaking apart the glucose and the fructose from the here, this is something everybody can do. But breaking apart the glucose and the galactose from lactose isn't necessarily something everybody can do after they've kind of post weaning. Other one, maltose. Maltose is just two glucoses connected together. We'll see glucose or maltose when we're looking at breakdown of starch, because as you break down starch, which are big chains of glucoses, you get them into smaller chains, including little double glucoses, which is a maltose. This is a good as time as any to just introduce some of the terminology often used in kind of some of the metabolism, like hydrolysis. People talk about hydrolysis, breaking things apart, hydrolysis. So if we look at a disaccharide, it's two sugars, and the little bridge between them is got this little oxygen. And so let's take a look at what some of the implications. If I have some sugar here and there's this hydroxyl hanging off, here's some other sugar and there's a hydroxyl hanging off. Again, there's lots more to these molecules than just the hydroxyl on these rings here, but that's all we're going to pay attention to right now. If I have two monosaccharides, and I'm going to bridge them here, what happens is the bridge ends up being like this, and we lose an H2O, right? Because two hydrogens and an oxygen get jettisoned as we bridge these two to make the disaccharide. So sometimes this process they call dehydration synthesis. Dehydration because it's losing a water, right? In synthesis, we're connecting these things. If I have a disaccharide and I want to break it apart into two separate things again, I have to use a water and add it in order to create two individual monosaccharides again. So to break this apart, we often call hydrolysis. Hydro, water, lysis, breaking apart, right? So the word hydrolysis just comes from the fact that you have to add a water molecule in order to complete this reaction, and it results in breaking apart this thing into separate pieces. So hydrolysis just means breaking apart and using a water molecule in the process. Okie dokie. So monosaccharides like glucose and fructose and ribose. Disaccharides, two of them connected together. Again, the most common one is sucrose, which is just the main transport plant sugar in plants. So that's the main sugar we eat, either cane sugar or beet sugar or any of those. Lactose is also a disaccharide milk sugar.
Good question.
Uh-huh.
Are disaccharides, is this just the examples? Are one of the monosaccharides always glucose, or can it be other things as well?
It can be whatever. It just means two simple sugars connected together. Yeah, though the physiologically relevant ones that I know about do have glucose in there, but I don't think that's not necessary.
Thanks.
So the last class of carbohydrates is when you have lots of simple sugars connected up in big long chains, and that would be a polysaccharide. So this is still carbohydrates. And the polysaccharides we're going to care about are just big long chains of glucoses. There's a few different polysaccharides that we are going to see in our class. Starch. Starch is one of the main storage carbohydrates in plants, right? That's what plants, if plants want to store away carbohydrates for the future, they create these big long chains of glucose known as starch. And then we kill the plant and we chew it up and we eat it. And then we have enzymes that break apart the starch to free up the individual glucoses, which we can then feed into our cellular respiration machinery and get lots of energy. That's why pasta is giving you energy, right? Like some wheat plant was storing away energy for a rainy day and then got turned into spaghetti. In animals, there's something very similar to starch. In fact, sometimes it's called animal starch. It's also a bunch of glucoses connected together, but rather than in a kind of a long chain, it's kind of more of a branching pattern. I shouldn't do that one. Glycogen, sometimes called animal starch. This is how your body stores away carbohydrate. Your liver cells have lots of glycogen stored in there. Like if your blood sugar starts dropping, you're going to turn on enzymes that are going to start breaking apart these bonds here in your stored glycogen in your liver and release it into your bloodstream to bring your blood sugar back up. Your muscle cells, particularly your fast twitch white muscle fibers, have lots of glycogen granules in there so they can release glucose really quickly and use it to make ATP quickly to, run your sprints and things like that. So there is a version of starch in our bodies and it's glycogen. And again, it's the same basic idea as the starch that we find in plants. It's just kind of more of a branching structure rather than a linear structure. There's one other polysaccharide that you need to know, and that's cellulose. So cellulose is a lot like starch, except it's got a different linkage between the glucoses. So maybe I'll do it as like a little Z or something. It's kind of more tightly wound up, and so these are still glucoses. But the big difference is this linkage here, this bond between the glucoses. Can't be broken by enzymes that we have. So cellulose, which is the thing that makes up the cell wall in a plant cell. It's one of the main structural things in a plant, right? It's wood, right? What is cellulose? It's like, wood. Wood is cellulose. So this often is what we call like the roughage or the fiber in our diet. Right? You can eat it. In fact, you do eat it. When you're eating plants, you're getting lots of cellulose. But because we do not have enzymes to break apart these individual glucoses, it just goes through our digestive tract, ends up in our intestines as kind of that roughage. We'll talk, when we get to the digestive system, we'll talk more about why that's a good thing. It kind of keeps things moving, keeps things hydrated, so your feces doesn't get overly compacted and things like that. Some of the bacteria in your intestines can break down cellulose, which is why the more beans you eat, the more you toot, right? Because the bacteria are breaking apart cellulose that you couldn't break down and making gas in your lower parts of your digestive tract. Right, there's lots of energy in cellulose. If you take wood and you put it on a campfire, it makes lots of heat and light. So obviously there's lots of energy stored in all those glucoses in that cellulose. But if you chew up a bunch of wood and swallow it, you're just going to poop out a bunch of splinters, right? So we do not have the enzymes to break apart and free up that energy that is stored in that glucose within the cellulose. So right, so cellulose is like starch, except it's locked, and we don't have the ability to unlock the glucoses from that structure. If you were a cow or a deer or some of these ungulates, these other creatures that rely on just eating plants for their main thing, then they have kind of these fancy ways. Like if you're a cow, you eat lots of grass, you get lots of cellulose, but then what you do is you kind of burp it up and you swallow it down, and you have different stomachs, and some of those stomachs are full of bacteria that can break down the cellulose. They're kind of like these fermentation chambers. Like when a cow is chewing its cud, it's basically bringing the food, the grass, down into chambers in their digestive tract where bacteria can break down the cellulose, release the glucose, and then they can burp it back up and swallow it back down into the other parts of the digestive system that can then utilize what the bacteria have freed up. So if you're a mammal, it's, and all you eat is grass, you have to have a fancier kind of digestive system that has symbiotic bacteria that allow you to work together to free up the glucose locked in the cellulose. So, right, but for humans, you basically are not going to get much out of eating grass. So, all right, any questions about cellulose? Okie dokie. All right, so that is carbohydrates. We can just go to lipids now, because lipids are also the same basic atoms, although in very different proportion. They're also kind of carbons, hydrogens, oxygens, but way more carbon and hydrogen. They tend to have these big, long hydrocarbon tails hanging off of them. So like the ratio, the proportion, these are much higher than the oxygens. As a rule, one of the main things you should think about when you hear something is a lipid, these are usually non-polar. We're going to see there is an exception. When we get to the phospholipids have non-polar and polar qualities, amphipathic molecules. But in general, maybe I should say in general. In general, non-polar. So like when we talk about steroid hormones being lipids, you've got to think non-polar, don't dissolve in water. So non-polar, let's talk about some of the basic kinds of lipids that we're going to see in the class. One of the main things are the triglycerides. A triglyceride is made from a glycerol, which is basically alcohol. And then 3 fatty acids hanging off the bottom. And each fatty acid is got like a carboxyl with these big hydrocarbon chains hanging off the bottom. And basically, this kind of bonds there. So you end up having, in kind of this kind of more cartoony way, you've got the glycerol as the kind of like attachment point. And then you have these fatty acid, these hydrocarbon tails kind of hanging off there. This is the fatty acid, and there's 3 of them going to be hanging off here. So triglyceride. Depending on the fatty acids, these triglycerides can have different properties. So let's talk about different kinds of fatty acids. So let's talk about saturated versus unsaturated fatty acids. If a fatty acid is got as many hydrogens as possible, and then it's called saturated. Saturated means it's fully saturated. It has as many hydrogens tacked onto that tail as you can put. Just in terms of its overall form, the tail tends to be straight. If you have an unsaturated fatty acid, that's when you actually have double bonds between the carbons. So wherever there is what's called a point of unsaturation, there's a double bond. You can't have as many hydrogens on there. So whenever you have these points of unsaturation, let me change the color. Right, we are now unsaturated because you don't have this total max of how many hydrogens you can put on this thing. What I've drawn here, I don't know if that's a real fatty acid. I'm kind of making this up. What really matters is this idea of it's not saturated, and wherever there's a point of unsaturation, wherever there's this double bond, the tail kinks. So when I draw one of these guys, it tends to look more like this if I'm drawing it as this kind of cartoon. And this is going to have implications for what happens when you make triglycerides with saturated fatty acids versus unsaturated fatty acids. So if I take triglyceride, and what we call a saturated fat versus an unsaturated fat. Again, the difference here is the types of fatty acids. If it's a saturated fat, it's got those fats that have the fully saturated with hydrogens, and the tails tend to be straight. These tend to be more what you find in animals. These tend to be animal fats. This is the fat you find in a person. Because the molecules can pack very nicely because the tails are straight, these tend to be solid at room temperature. The unsaturated fats, these tend to be found in plants. These tend to be plant. Because they don't pack so well, because they have those bent up tails on the fatty acids, these tend to be liquid at room temperature. What's another word we often use for these unsaturated triglycerides, unsaturated fats? Liquid.
Good fat.
What's that?
The good fats.
Yeah, in general, the unsaturated ones are correlated with better cardiovascular health for sure. But what's another word for I just said it. Fats that are liquid at room temperature.
Oils.
Oils.
And oil.
These tend to be oils. The only difference between an oil and in fat is, the melting point. In fact, if there's only one point of unsaturation, then it would be called a monounsaturated fat. Olive oil is a monounsaturated fat. Olive oil is liquid at room temperature, but if you put it in the fridge, it gets solid pretty easy. And then it liquefies again when you bring it out. Polyunsaturated fats like, corn oil or safflower, those stay liquid even when you put them in the refrigerator. So polyunsaturated just means lots of points of unsaturation, which would be like really bent up like this. Monounsaturated, if there's like 1 point of unsaturation. Like someone was just mentioning, in general, unsaturated fats in your diet tend to be correlated with better cardiovascular health than eating lots of saturated fats, lots of animal fats. Which led to some weird, we should, before we leave fats, we should mention the idea of hydrogenation and trans fats. So for a couple of reasons. One is this idea that unsaturated fats are healthier, and also saturated fats are just kind of easier to work with. They're kind of creamy. You can spread them like butter. Or, think about if you make peanut butter and you grind up a bunch of peanuts. If you've got like natural peanut butter, it's a pain in the butt because it's like the oil separates from the peanuts and you got to mix it up before you actually use it. So it would be nice to have these plant oils, but have them like solid at room temperature so they could be creamy. So what people did was they had this process known as hydrogenation. They would take hydrogens and slam them on to the tails in the plant oils. So hydrogenation. Let me spell that right. I'm writing too. Let me slow down my writing. Hydrogenation. Basically, this chemical process that straightened out the tails, turned corn oil into margarine, right? Or took peanut butter that had separated into oil and peanut solids into just Skippy and Jif, creamy stuff that never separated. So, it was when I was growing up, there was this kind of now quaint thought that margarine is better for you because it's made from plant oils, even though it kind of looks like butter. But what they realized was in that process of hydrogenating the fats, that they were creating these weird bends and kinks in the tails that were not things that your digestive machinery could actually deal with. So the idea of trans fats, right? If I have some like tail that's bent in different ways, there's trans versus cis. So trans fats just would mean something was bent rather than bent like this way, things were bent like the opposite way. So you created these fatty acids that were bent in ways and had little kinks that your enzymes did not have a way to deal with properly. And it turns out when they looked at this more closely, that even though these were unsaturated, they were worse, as bad or worse for you than just eating saturated fats, that you would be way better off just eating butter than eating margarine. So it was kind of hilarious. So at this point, trans fats are no longer, found in processed food. The process of trying to hydrogenate things is useful. It makes liquid things creamy. It's solid at room temperature. It also makes them stay from going rancid. Those points of unsaturation are places where things tend to break down more. Like a saturated fat is more likely to not break down and not go rancid. But they've obviously figured out ways to process things to not create these trans fats the way they used to, because it turns out they're not good for you. So trans fats, they are artificially created in that process of hydrogenation, trying to turn oils into kind of solid at room temperature. Another version of this triglyceride that's not a triglyceride is the phospholipid. We should talk about those. So if you take that glycerol, let's go back to here. If you take the glycerol and you put on 2 fatty acid tails, but on that third thing, you actually put this polar phosphorus containing group, then we are going to now have a phospholipid, which is the essential component to make up cell membrane. So let's talk about phospholipids. So this has part of it that is polar and likes to live in a polar world. Another part that is nonpolar and likes to interact in a nonpolar world. The official word for molecules like this are amphipathic. I think kind of like amphibians live in the water and in the land, like a frog, you know. Amphipathic molecules have part that live in polar, part that live in non-polar, like to interact. If you take these and you just put them in water and you shake them up, they will just naturally arrange themselves where the hydrophobic tails all go towards each other and the hydrophilic heads go out towards the water. So we get the phospholipid bilayer, which should not look surprising. This is something you've seen in your Bio 110. It's the way that a cell membrane is formed. Phospholipid bilayer. Again, the polar heads. You know, and I have all my H2Os. So my cell membrane automatically just configures itself. You don't have to do anything. If you just take phospholipids and you shake them up in water, they will create these little vesicles with this phospholipid bilayer membrane surrounding them. Again, they are just held together purely by the hydrophobic hydrophilic interactions. Maybe I'll write that specifically. There, grab it. Right, there's no actual chemical bonding between one phospholipid and another phospholipid. It's just the polar heads want to be out towards the water, the tails want to get away from the water, and go turn in towards each other. So this is a cell, this is what your cell membranes are made out of. We're going to spend lots of time in this class, so over the next week, looking at cell membranes, but let's spend a few more moments thinking about this. And I should mention the classic phospholipid that makes up your cells, you've probably heard of. It's like called phosphatidylcholine. It's just lecithin. You can just go to like a health food store and buy lecithin in little gel caps. It's just this oily stuff. But if you take that lecithin, that oily stuff, and you mix it up, it becomes this. This is also how detergents work. We'll talk about this more later. But if you have oil on, maybe I should just do this really briefly. Let's say here is your plate, and it's got grease all over it because you were just eating French fries, and you want to wash this plate though, and it's all full of oil from your French fries. And you are pouring water down from here's your sink, and you're pouring water. The water is not going to take the oil off the plate down the drain. The water is just going to skim across the oil on your plate, right? Because hydrophobic. So just taking a greasy plate and rubbing the oil underwater isn't going to do anything. Instead, you have your detergent like Palmolive or Dove or whatever you're using is another version of these amphipathic molecules, these little and what happens is when you add the soap, the detergent to this, then the little oils get surrounded. The non-polar tails go into the oil drop. The polar heads go out towards the water. And now this little thing can get swept up in the stream and goes down, and you eventually get the oil off of your plate. We're going to see a similar thing like in digestion when we look at how bile helps break down fats in your digestive tract. Bile is another little molecule like this amphipathic molecule that's partly polar, partly non-polar. But just to clean greasy dishes, you need amphipathic molecules, or else the water would never be able to carry away the oils, because you have to be able to get some intermediary between the oil, the non-polar and the polar, in order to get the oil droplets up into the water stream. Doobie-doo. All right, so back to our phospholipid bilayer. Let's talk a little bit about, actually, we'll come back to this in another moment. Let's erase this. Let's talk about cholesterol. Cholesterol is another lipid. It's like 4 interlocking carbon rings. Don't worry about the actual form of it. You're not going to have to recognize a cholesterol molecule, but you should know that cholesterol is a lipid. Cholesterol is also it's going to be an important actual part of cell membranes in a way that we'll talk about in a little bit. Cholesterol is also the starting point to make steroid hormones. Right, so what's important here is, since steroids come from cholesterol and cholesterol is a lipid, steroid hormones are nonpolar, which means we're going to need to deal with them differently than we're going to deal with other things in your body that dissolve in water. Let's go back to this cell membrane. So there's also little cholesterols that are kind of stuck in between the tails here. So when we look at a phospholipid bilayer here, it's a phospholipids as well as, and again, the cholesterols just stay put because they're nonpolar. They want to be in that nonpolar region amongst the tails, right? There's nothing that's going to get them to want to leave, because if they start moving out towards the water, they just get squeezed back into the nonpolar region. So while we're going to develop this idea of a phospholipid bilayer in much more detail and add in all of the membrane proteins. But for right now, let's just stick with this, and then let's talk about permeability of a cell membrane, just based on what we have here. Make it darker. Right, we're going to be spending a lot of time talking about what can move in and out of a cell and how it's controlled. So before we get into all of that, let's just look at the basic properties of a cell membrane as a phospholipid bilayer, and just what can naturally move across a cell membrane without any help at all. So assuming that you have something that makes it up to one side of a cell membrane, what is going to determine whether or not it can make it to the other side?
Permeability?
But what's going to, that by definition, it's permeability, but what is going to determine whether or not it's permeable?
Whether it's polar or non-polar?
And what about what kind of, in which way? Be more explicit. What kind of things will be able to easily just move from one side to the other?
Smaller molecules.
Non-polar small molecules.
So non-polar. Things that are non-polar can cross without any kind of help, right? It doesn't matter their size, right? Size doesn't matter here. It just matters if you're non-polar and you're here, you can just drift into here and drift across and end up on the other side. You know, once you're on the other side, something's going to have to pick you up and carry you away. But getting across the membrane, if you're non-polar, it just happens. Right? Because the membrane, the actual kind of expanse from one side to the other is this non-polar region of these hydrophobic tails. So things that are non-polar don't need help to cross a membrane. If you're a steroid hormone and you make it to the cell membrane, you just drift across and end up inside the cell without any help. Drugs that are non-polar can just get into the membrane without any help. What about polar things? Can a polar molecule, is a polar molecule going to be permeable?
Yes.
Why?
Well, isn't there going to be some sort of issue with the polar head? So isn't there going to be some sort of like repulsion that could potentially happen?
Right, so if I'm a polar molecule, let's say this is sugar or something, or glucose or whatever, you know, with my pluses and minuses, this is some polar molecule. as soon as I get around here, I'm going to want to just stay attracted to all of the other polar molecules. There's no reason that I'm going to go into this non-polar nether zone of no charge. So non-polar molecules, pass through the membrane easily. Polar molecules, in general, not permeable. Because if you're polar, you're going to want to stay in your polar milieu, do your hydrophobic, do your electrostatic attraction dance with the other water molecules and stuff. So if you're on one side of the membrane or the other side of the membrane, you're going to keep staying on that side because that's where all the attraction is, keeping you pulled and stayed there. If you start getting close, I'm going to erase this thing to give myself more room here. Right, I have some little sugar molecule here with its kind of polar nature to it. It's going to want to just keep doing its attraction to the water molecules. If it ends up getting up close up to here, it's like, what's there? There's nothing interesting there. It's going to stay with the water. So in general, polar molecules do not penetrate a membrane without help.
I have a question. So does the polar head of the phospholipid not play into that? It's just the.
But we're talking about being able to cross the membrane, right? The polar head could be attractive to this, but
Right.
then continuing on into this region of non-polar tails, that's not interesting at all. And this polar molecule is just going to stay on one side. So what's relevant here is that the main expanse of the membrane is this non-polar region that's constructed by these non-polar hydrophobic tails. So again, this is this big non-polar region here. So polar things in general do not go across. There is an exception. Except.
Ion?
No, we'll get to ions if really small. So for instance, water. A water molecule. A water molecule is obviously polar. But if water couldn't cross a membrane, then we wouldn't have to worry about osmosis or anything. So obviously water does cross membranes. The reason they think that water can cross is that really small things are just moving so fast. They have a lot of momentum, right? Or for certain, momentum is mass times velocity. So if your mass is really small, you have a really fast velocity. So they manage to there's moving so fast, they get in here, and by the time they realize, I'd rather not be here, I'd rather be attracted to other polar things, boom, they're on the other side. So people talk about it kind of like a bullet or something. Like even you're shooting a bullet through there, even if the bullet actually would rather stay here, by the time it realizes that it's more attracted here, it finds itself on the other side. So really small polar molecules can pass through the membrane. Maybe I should draw this in a prettier way. So like water molecules can go across. Things that we're going to see in our lab. Another one like urea. Urea is a polar molecule, but actually that has some permeability across the membrane, which is actually going to be important when you're coming up with your hypotheses for a lab next week. I should mention that the amount of cholesterol, I talked about cholesterol being important here inside the membrane. The more cholesterol that is stuck in there, the more packed in the tails are and the less permeable the cell is to water. So you actually do have some cells that really don't let water across. They're more specialized and they have like lots of cholesterol packed in there to change the permeability of the membrane, just by making the tails much more packed and making it a lot harder for things to sneak in between. Somebody mentioned ions, so let me talk about that briefly. So what somebody might think like, oh, an ion, it's polar, it's charged, but it's really small. It's just an atom. So can that make it through? Like, what do you think?
Doesn't it depend on if it's a positive or negative charge? Or no?
No, it wouldn't, because if it's positive or negative, it's still gonna be polar and want to interact with the polar water molecules. So whether it's positive or negative wouldn't matter there. What does matter is that it's really highly, highly charged. It's not just like local charge imbalances like sugar or something. If I have an ion, let's say a sodium or a potassium or a chlorine or whatever, it's super positive. And what happens is the water molecules around it start arranging themselves in almost like a crystal. They talk about it being like ice. So you get this kind of hydration sphere. You know, basically, I'm going to say like ice. The water molecules are rigidly sucked in and hold this form around that ion. So effectively, or I should say like a snowball. So rather than having an individual little polar atom, effectively you've got this big snowball that is all gathered around that. So when we go to this picture over here, well, you know, my little, what looked like a little sodium is not a little sodium. It's actually, and it has all the H2Os around it in this big sphere. So it actually acts as this big thing that cannot cross the membrane on its own. It can only cross using specialized membrane channels that we're going to talk about in detail next week. So ions cannot cross on their own without help. Cannot cross membrane without help. And again, typically it's going to be these specialized membrane proteins, these ion channels that we're going to be talking about in detail. So definitely membrane permeability is going to be an important theme in our class because it's the boundary between you and not you, right? It's what's inside the cell and outside the cell. So understanding what can cross easily, non-polar things, what can't cross polar things, unless really small. We're going to spend a lot of time talking about the specialized membrane proteins that then help control the passage of things in and out of the cells. But membrane permeability will be an important ongoing theme in our semester. Trying to think if there's any other. I think that's the lipids. I wish we haven't quite got to the proteins yet. So any questions about lipids?
I have a question about the sphere of hydration you just described. Or actually, two questions. So question number one, they tend to form that because of how charged
How
the
strongly
ion is in
charged
the
they
first
are.
place.
Yes.
And question number two, same goes for cations, for negative ions, negatively charged
Exactly.
ions.
It doesn't matter whether it's a cation or an anion.
Sorry,
it's going to be the
anion.
same thing.
Okay, thanks.
Yeah. So ions, both cat and an, cannot cross without help of specialized membrane proteins. Right, and that the whole thing around signaling, electrical signaling is going to be about opening and closing ion channels and letting ions flow. And as we talked about, flowing ions, flowing charges, electrical currents, that's going to be the electrical currents of your nervous system and creating voltage changes across the membrane and things like that. So by allowing them to enter cells or not, that is going to be controlling kind of the electrical activity of your neurons and other cells, excitable cells like muscle cells as well. Just to kind of review what we have been talking about. Let's see. I'm going to share. These are basically PowerPoints from your textbook. It's the stuff that's uploaded just on molecules. Just to kind of review this, when we're talking about lipids and the fatty acids, these are fatty acids. Like I said, one side has that little carboxyl, and then you've got these big long hydrocarbon chains. A saturated fat has as many as possible hydrogens as possible on there, and monounsaturated has like 1 point of unsaturation. Here we see many points of unsaturated, a polyunsaturated fat. This picture is not giving you the whole story, unfortunately, because it doesn't show the bending. Like this would be like straight, this would be bent, this would have lots of bends. In fact, let me go. If I share, right, this is like a pic, just some stuff. If you like look at Google unsaturated fats, this is more what I'm talking about. A saturated fat that's fully saturated with hydrogens, which we tend to find more in animal fats. That's why they got like the bacon here. As opposed to unsaturated fats, those points of unsaturation cause bends in that chain, in the hydrocarbon chain. So just kind of being clear about the unsaturated fats tend to be, have the kinks. Unsaturated fats tend to be the oils liquid at room temperature. Okay, back to the PowerPoint. And when we want to make a triglyceride, it's just taking that glycerol and adding in 3 fatty acids. And again, I wish they had them all go in the same direction, but for whatever reason, they're doing it this way. Steroids, again, I was mentioning the 4 interlocking rings. You don't got to worry about the detail. Eicosanoids, that's actually another, you should know that word, eicosanoid. Let me highlight it just so. The reason why you should know the word eicosanoid is that if you read that some molecule is an eicosanoid, you should know that it is lipid and therefore is, nonpolar. So there are going to be important signaling molecules in the body that are eicosanoids. So that's why you should, just the same way the word steroids are also lipids. Eicosanoids are another class of molecules that are lipids. And beyond knowing that they're lipids, you don't need to know anything else. You just should recognize if you see that word, oh, I'm looking at a lipid. Phospholipids, like I mentioned, have the 2 fatty acids on the glycerol, but then that polar phosphorus containing group there, which is going to be critical into their amphipathic properties. And here we have monosaccharides, we're in the carbohydrates, simple sugars. Again, in water, they tend to form this ring form. Disaccharides, sucrose, maltose, lactose, we were talking about. These are if you connect 2 simple sugars into a disaccharide. A participant requests that live transcription be enabled. I don't even know what live transcription is. Can someone tell me what that it means?
It's like subtitles, I believe.
Oh, sure. All right, it is on. That's cool. I didn't even know that existed. That must be kind of one of the more recent things. Oh, now it's bizarre. I'm like seeing my words showing up at the bottom of my screen. It's a little weird. All right. Polysaccharides, again, the starch, cellulose, we've talked about. Glycogen is like the animal starch. Chitin is similar. It's actually I think chitin has a nitrogen in it as well, but it is the main structural carbohydrate in like insects and crustaceans. It's like, this shell of a shrimp. I'm pretty sure the dude who initially synthesized LSD, his dissertation work in grad school was actually figuring out the structure of chitin. I mean, that was he was into natural product chemistry. That was part of what led him down the road to finding different things made out of these molecules made from fungi and stuff. So proteins and amino acids. This is, I'm looking at the time. We actually have plenty of time. We've got like over 2 hours left in our class. The jumping lab stuff that we're going to be doing is not going to take anywhere near that. So I think in order to keep the class flowing, we actually have less days this semester than we had last semester just because of holidays and stuff. So I think I am going to spend hopefully 15 minutes just finishing up our chemistry review just by talking about proteins and being done with it. And then when we regroup on Tuesday for lecture, we'll just fully be in physiology. I mean, it's going to still be chemistry of cells. It's going to be membrane proteins and things, but at least we'll be talking about cells rather than talking about just pure chemistry. So I am going to just finish up our chemistry review by talking about proteins. You know, RNA and DNA and all that is also important. But for our class, we're not going to really be, I'm assuming you understand the basics of DNA and how it encodes genetic information, and there's messenger RNA and ribosomal RNA and transfer RNA. It's all stuff you did in your Bio 110. We talked already about NADH. We've talked about ATP. So there are going to be nucleotide containing molecules that we're going to be using for, particularly for carrying energy. But for the most part, we are not going to be going deep into that. But we are going to be going really deep into proteins. So let us introduce the protein. So a protein is basically a chain of amino acids. Lots of different amino acids. There's actually 20 amino acids that are naturally found in our bodies and our proteins. You know, there's more amino acids than that, but those are the amino acids in our proteins, right? Those codons in the DNA code for one of these 20 amino acids. So that's how your cells know which amino acids to hook up in what order to make a particular protein. For this to make sense, we probably need to go and define an amino acid. Amino means this little amine group. That's the amino, right? It's just a nitrogen is an amino. The acid is a carboxyl. So the carboxyl is the acid. So the amino acid, and then there's this side chain. This is what's different on every, any of these 20 different amino acids. They all have unique side chains, and the side chains give any particular amino acid specific chemical properties. You know, some are non-polar, some are polar, some are polar positive, some are polar negative. You know, some make ionic bonds, meaning not just charge imbalance, but are going to give up or accept an electron to make an ionic bond. So different amino acids like to participate in very different kinds of chemical attractions. That's going to be critical in understanding how like receptor binding works when we get to like, what does it mean for serotonin to bind a receptor on your neuron? It's going to have everything to do with what amino acids make up that serotonin receptor and what kind of chemical interactions those amino acids like to make. And like I said, we'll get more into this in detail. But again, some are nonpolar, some are polar, some make ionic bonds, positive or negative ionic bonds by either accepting or donating an electron. So lots of different amino acids. You connect them up, amino to acid with peptide bonds. And you don't need to know the details of exactly that. These are just called peptide bonds. A typical protein is made up of thousands or tens of thousands of these amino acids connected up together. They're huge. If less than 100 amino acids, then you typically call it a peptide rather than a protein. There's a lot of really important biologically relevant peptides, a lot of hormones like insulin. Insulin is 37 amino acids hooked up, and just the right ones in the right order, folded in the right way. That is insulin, your hormone. A lot of the natural opioids, the things that bind at your opioid receptors, are just little peptides. So a lot of really important biologically active things are little chains of amino acids. The bigger ones, the ones that we're going to talk about in more detail now, tend to be much larger, like hundreds, thousands, tens of thousands of amino acids connected up together in these huge molecules, complicated molecules. What I've drawn here, this is called primary structure. It's just kind of the order of the amino acids. And again, what defines which amino gets hooked up in which order? How does a cell know how to put this thing, assemble this thing?
The enzyme?
No.
The tRNA, the codons.
The codons, right? Remember the DNA in your nucleus has those codons, those three base pair codes that define. If you were in Bio 110, you took like the felt, right? Each amino acid was made out of felt, and you would figure out what is the 3-letter code for that amino acid. And then, okay, let's take that piece of felt and put that one up next. So you've got your DNA, which then gets transcribed into the messenger RNA and gets edited and finally goes to the ribosome. And then the transfer RNA brings up amino acids, with the anti-codon that is going to match the codon. So we assemble the correct amino acid in order at the ribosome to make the protein. In actual cells, there's all sorts of different ways to edit the messenger RNA, and there's post-transcriptional like modifications once you're done assembling things. But let's keep it simple right now. So primary is just this big long snake of amino acids, but that is not where it stops. So what happens is from here is you get hydrogen bonding between different amino acids in the chain. There's two main forms at this hydrogen. So we're now going to go to secondary. Secondary structure. There's two things that happen. One is where every third amino acid hydrogen bonds with the one above it, and you get these helices. They're called alpha helices. If I'm going to draw this better, it's like 1, 2, 3. So every third one starts making these hydrogen bonds. So it stabilizes as these helices, these spirals, due to the hydrogen bonding between every third amino acid. The other possibility is sometimes they kind of create what are called beta pleated sheets. So instead of making spirals, they kind of make these big, more sheets like this. So this is the secondary structure. You get hydrogen bonding between adjacent between amino acids within the same big molecule, within the same big protein. And it takes the form either of these big, like pleat-like folds, the beta pleated sheets, or else these kind of tight spirals, the alpha helices. Then we get more complicated stuff going on, basically like kind of hydrophobic, hydrophilic interactions. More hydrophobic parts of this overall thing are going to want to kind of move towards the inside. More hydrophilic stuff wants to be more out towards where the water is. There is also there's often different proteins that actually help with the folding to make it get to the exact form. But so the next we're going to get our third or tertiary structure. This is going to be hydrophobic, hydrophilic, plus I'll say plus more, because sometimes it's actually assisted by special molecules that help with the folding. There's also these little stabilizing like disulfide bridges and things. But basically you get this big complicated thing. Parts of it are sheets, parts of it are so I drew it looking like a big messy thing of spaghetti because that's what they look like. Exactly what the form looks like is going to depend on its function. Like ones that are going to act as an enzyme are going to be kind of more globular. Ones that act more as a structural thing might be more fibrous to make more structural fibers and things. Many of the proteins are functional at this level. If this is some enzyme, it might be, this is a little pocket right here. That's the active site that has, it's folded in just the right way to make this pocket just the right shape to fit the sugar, to break the sugar apart or something, right? This is why we can, when we talk about like pH, denaturing enzymes, the idea of denaturing an enzyme, denature means lose its shape. Right, pH is adding more or less H pluses around. If I start adding a whole bunch more H pluses around, that's going to totally change the polar qualities, and it's going to, like, things are either going to want to pull out or squeeze in depending on whether they are plus or minus. So this whole thing is going to change its shape. So changing pH is going to denature a protein, cause it to lose its shape. And if it's an enzyme, probably cause it to lose its function. While we're here, denaturing. So pH can denature a protein. What other things can denature a protein?
Heat.
Heat, totally.
Temperature.
Yeah, so temperature. So again, if we kind of go back, temperature, imagine all of these things are wiggling. All of us, before you know it, they're going to get all tangled up in a big irretrievable mess. So like a classic example, you take eggs. Egg white is albumin, it's just a protein. And you heat it up and you entangle all of those chains of amino acids. And before you know it, you just get that big solid white mass that is a cooked egg white. If we think about pH, you can cook with pH too, right? If you want to cook a fish, you can heat it up and denature it, turn that kind of translucent fish flesh into white flaky fish, or you can put it in some lime juice and make ceviche. That works nice too, to make a delicious like cooked fish, denaturing the fish flesh. What other things can denature an enzyme? Going back to the egg white, you can take an egg white and you can boil it, turn it to make it into a solid white. How else can you make an egg white into some solid white?
By whipping it?
By whipping it. You can make a meringue. So you're just mechanical. I mean, literally, you're just beating the crap out of it and the little chains are getting all moved around and getting tangled up. And before you know it, they're a big tangled mess. And you got, kind of stiff meringue peaks to put on top of your lemon pie. So it's worth remembering what kind of things denature, particularly heat and pH in the context of our bodies, because particularly enzymes are going to lose their function if they lose their shape. Some of the people in the warm-up quiz talked about kind of the active site. It's, you want to make sure you remember, like an enzyme works because it has a particular active site that will specifically bind the substrate. And if something denatures it, changes the shape of this active site, it's no longer going to be able to bind its substrate and the enzyme will no longer function. So, I mean, it's one of the many reasons that we need to maintain our temperature homeostasis, our pH homeostasis in our body, is so all the enzymes are in their happy place to be able to catalyze all the metabolic pathways. There's one more level of structure that's optional. Quaternary structure. This is two or more tertiary structures to make functional protein. We'll see a bunch of cases of this, like hemoglobin in your blood cells that carries oxygen are 4 globin subunits that go together. Myosin in the contractile protein in your muscles have light chains and heavy chains. The antibodies that are part of your immune system. These are all examples of complicated proteins that have more than one third order subunit to put together to make the actual functional protein. So I am now going to so now we're back here on your the PowerPoint that's part of from your book. Just kind of reiterating, primary structure is just the sequence of amino acids connected together. And like I said, it might be tens of thousands. Secondary structure is either alpha helices or beta pleated sheets. tertiary structure is where you start getting much more elaborate folding, usually due to this hydrophobic, hydrophilic interactions, as well as other things going on. And again, many proteins are done at this point. But then we do have examples that I just mentioned where you have the quaternary structure, where you need more than two or more of these third order subunits to make the functional protein. Nucleotides, like I said, main nucleotide based things that we care about are ATP and ADP, FAD, cyclic, actually I should mention cyclic AMP. Let's leave it. Let's do that real briefly. So before I do that, any questions about proteins? All right. So let's finish up with nucleotide based things. DNA, RNA, everybody, hopefully everybody is pretty clear about those. A, T, C, G for DNA, AU versus CG for RNA. The ones that will be used more in our class are ATP, the currency of energy in the body. And again, we're going to see ATP breaking apart into ADP plus phosphate, plus energy. And again, ADP, just diphosphate, it's got 2 phosphates. ATP, triphosphate, it's got 3 phosphates. A word you should know is exergonic. Releases energy. So this, what I've drawn here is an example of an exergonic reaction. Breaking apart ATP into ADP plus phosphate releases energy, which then can be captured to do other things. This is in contrast to endergonic. There are endergonic reactions that need to happen in your body. And so the way that usually happens is you couple these. We're going to see lots of examples where we tie the hydrolysis of ATP into some action that would not normally happen on its own without an energy input. So we're going to see a lot of cases where we use ATP to provide the energy for some reaction that needs energy to run. That's what I mean by coupled. They are kind of linked together. So you use, it's kind of like putting the gas in your tank, right? A car isn't normally going to roll from your home to campus, but if you couple the combustion of the gasoline to the turning of your axle, you can actually use the energy producing of burning gasoline to roll your car from your home to the campus, which takes energy. So I don't know if that's helpful. Again, the other one, nucleotide things I talked about, like NADH, which is NAD plus 2 electrons plus H plus. This is again my high energy electron carrier. This is gonna show up playing a center role when we look at cellular respiration as a way to transfer energy from one place to another. Basically, NADH has a lot of energy because the electrons are held in this really high energy configuration. And when it donates those electrons, again, like I said, it's similar to you're holding like a book up near the ceiling and you let it drop and catch it near the floor. As it's falling, you can capture that energy to do something. What atom is the equivalent of like catching the book at the floor? What's the thing that will accept electrons at the kind of lowest energy level? Does anybody know? Like if I have electrons in some molecule and I want to have some redox reaction where I'm giving them to something else to accept them, I need to have something that is able to like catch them at a lower energy level and hold them there. All remember electronegativity and all that stuff. That's where like oxygen is going to come into play. Oxygen's like claim to fame in these reactions is it can catch electrons at kind of this lowest level energy. So even if you're holding them relatively low, there's probably still some place for them to drop to finally get loaded onto the oxygen. So oxygen, when we talk about like why you need to breathe oxygen, why do you die if somebody like, chokes you to death or something? You're not getting any oxygen, but why does that matter? It's like ultimately all the machinery of cellular respiration is going to stop if you don't have this final electron acceptor that accepts the electrons after all these redox reactions where the electrons have been dumped from one to another molecule in this process. So we'll meet oxygen as kind of the final electron acceptor when we look at cellular respiration next week. The other nucleotides that you are going to need to know. So this is adenosine monophosphate or guanine monophosphate, and this means cyclic. So this is cyclic adenosine monophosphate, but usually just called cyclic AMP. This is how it's known, if you're just kind of talking about it. These have a very different role from everything we've been talking about. These are important signaling molecules. They are not about providing energy or anything else like that. However, when we get to signal transduction in the coming weeks and talk about, how, a neurotransmitter binds on a neuron and then something happens, that process of something happens involves cyclic AMP or cyclic GMP. These are also the targets of all sorts of drugs because these are important signaling molecules, especially within the cell. I should say important within the cell. One of the more popular drugs like Viagra works by modulating amounts of cyclic GMP in cells that help prolong erections. So these are really important in how cells do their thing. And we will be spending quite a bit of time seeing processes that are facilitated by cyclic AMP and cyclic GMP. Cyclic GMP is at the core of how your rods and cones convert light into electrical signals, so you can actually see your screen right now. So these aren't just like, oh, by the way, there's also this other thing. That these are going to be important. They'll be your friends. By the end of the semester, you'll definitely see cyclic AMP and cyclic GMP and think of them as just another fundamental molecule in cells that are doing super important stuff. All right. I talked more than I thought. Any questions about any of this? Right, we're done with the chem review. if hopefully you were able to kind of get a sense of what you're still comfortable with, what you need to kind of go back and review a little more on your own. All of the stuff that I've been mentioning this past 2 weeks is going to be kind of foundational for the coming 4 months, or I guess at this point, 3.5 months. So if some of these concepts are still confusing you or a little fuzzy, please, like early on, right now is when you should try to get them solid. I'm going to be in office hours from 12:30 to 1:30. You can ask me questions there. You can email me things. You can go on to YouTube things. You can go and look at Khan Academy. Go to your old textbook. Go to tutors. In fact, we are going to have tutoring. I'll talk about that a little later. But please do not let the chemistry just like sit and kind of don't think like, okay, that was a little confusing, but now we're getting to the cell. It's like, if this part isn't making sense, the cell is just going to get even more and more confusing, because we are going to just be bringing in all of these concepts into understanding how a cell works. So, all right.